Hello, everyone. Welcome to this presentation. My name is Nikita, and today I'm going to present the PNI's proprietary lipids, the lipid nanoparticles that we have formulated from using them, and how we have used them to develop the proof of concept data for cell therapy, protein replacement therapy, and vaccine applications. Now, to begin with, first I would like to explain what a lipid nanoparticle is. So, a lipid nanoparticle is simply a drug delivery vehicle where the nucleic acid is the drug. It consists of four major components. So, the first one is ionizable cationic lipid. And if the ionizable cationic lipid is of the desirable pKa, it becomes positively charged when it comes in contact with the formulation buffer, and it can easily bind with the nucleic acid, which is negatively charged. So the other function would be if it is in the endosome, it becomes positively charged and it aids in the endosomal release of the nucleic acid into the cytoplasm. And it is also neutral when it's at the physiological pH. There are other components, that is cholesterol, helper lipid, stabilizer. Each of these components have their own important roles, either in terms of the self-assembly or in terms of stabilizing these particles. Now, moving on to the first application, that is the cell therapy. I would like to first just give a brief overview of what it is. Now, in cell therapy, what happens is the blood is drawn from the patient and the T cells are isolated from the blood. And these T cells are then transfected with the lipid nanoparticles that is carrying, say, for example, CAR mRNA. And hence, these CAR mRNA are then expressed on the surface of the T cells and are re-injected into the patient. So this cycle overall kind of compiles the cell therapy, where the cell itself is the drug. And in this scenario, lipid nanoparticles have accelerated the cell therapy manufacturing workflows. Now, at Precision NanoSystems, we have used eGFP mRNA to show the proof of concept, where the GFP is the reporter gene. And you can see that we have used the PNI proprietary lipids, and we have formulated lipid nanoparticles in encapsulated GFP mRNA. And I have shown three sets of data. That is, first one is the GFP MFI, and the other one is transfection efficiency, which shows the percent of GFP of all live T cells. And the third one is the viability. And in all these cases, we have compared them against the benchmark, that is MC3 lipid. As you all know, that Onpattro was got their FDA approval in 2018, where in the formulation, one of the ionizable lipid component is MC3 lipid. Hence, for all the experiments that we're doing in-house, we are always comparing the potency of the PNI proprietary lipids against the MC3 lipid. Now, you can see that in all these cases, the PNI lipids actually outperform the MC3 lipid. And in terms of viability, there were no cell deaths. The viability was excellent. In continuation with these data, there are other sets of lipids. Now, again, these are only representative examples. We do have a lot of lipids that have outperformed MC3. Now, these are, you can see that IL-6, 7, 8, 9, 10, they have all outperformed MC3 lipid. Now, the take-home lesson from here is that we have found that the ionizable lipid has to be in the pK of the ionizable lipid has to be in that sweet spot, as highlighted in this graph over here. And if it's in that particular range, then we can see some promising potency in the lipids for delivery of the RNA for cell therapy. But if the pK is very high, then we see significant cell deaths. And if the pK is too low, then there is no cell deaths, but there is no transfection as well. And we also had quite good success with the CAR-T therapy using PNI lipids. And I would recommend you to attend Rika Gezi's presentation for details. Her poster number is 473. Moving on to the next application, that is the protein replacement therapy. We have used erythropoietin as the protein of interest. Now, if you look at the anemic patients who became anemic by kidney disease or cancer therapy, they are usually treated with the exogenous erythropoietin. Basically, erythropoietin is just injected directly into them. Now, what people have done is that people have encapsulated the EPO mRNA, which encodes for erythropoietin protein, in the lipid nanoparticles, and they have injected that intravenously, where these lipid nanoparticles then delivers the mRNA to liver cells, and it expresses EPO protein that is stimulates red blood cell production. And just as a proof of concept, again, we have indeed used EPO mRNA. We have encapsulated that using the PNI proprietary lipids. And you can see that in there are quite a good number of lipids where we have seen that they have either expressed as good as or even higher than what MC3 has expressed in terms of erythropoietin. And we can say that that in almost all of these cases, functional erythropoietin was expressed. The reason why I say functional is that these erythropoietins, actually the hematocrit analysis was done after 7 days of the EPO-LNP injection. And we found that these were able to increase the 40% hematocrit levels across all the batches of IL-1. So this is just one of the examples, but we have seen that in most of the cases, we have seen increase in the hematocrit analysis. Now, moving on to the next application, that is the vaccine applications. Now, you have already heard about BioNTech and Moderna mRNA vaccine. Here, I'm going to focus on saRNA vaccines. Now, what is the difference between the saRNA and mRNA? It is that saRNA is able to, or self-amplifying RNA is able to replicate itself, as a result of which the advantage is that we can get done what we want to get, we can get done with even low dose of saRNA for the vaccine purpose. And our very first set of data was generated in collaboration with Imperial College London in Professor Robin Shattuck and Anna Blackney's group. And from Precision NanoSystem, we had shipped few lipid mixes to them, and they had formulated lipid nanoparticles where they encapsulated hemagglutinin saRNA. And you can see that significant antibody response was seen after the mice groups were boosted. I would like to draw your attention to IL-1, where you can see that the amount of the antibody that was response that we got in that case was in the order of 10 to the power 8. And when these groups were challenged at 6th week, we saw that the mice group that were able to generate significant amount of antibody, they were really able to cope up with the challenge study, and there was no loss in the body weight of the mice at all. However, in the cases where there was less amount of the antibodies developed, you could see that there was significant loss of the body weight. However, at the end, they were starting to recover. Now, we also had some success with the SARS-CoV-2 vaccine, and I would recommend you to attend Srinivas's presentation. His poster number is 477. To sum up, we have shown you the proof of concept data for where we have used PNI, the lipid nanoparticles that was formulated using PNI proprietary lipids in the field of cell therapy, protein replacement therapy, and vaccine applications. We do have a big library of lipid with the diversity of pKa in there. So basically, depending upon the type of application, what we can do is we can choose a certain set of lipids and screen them for that application. Once we find the hit, for example, the green ones that you find the hit, and then we can take them further down the road until we find that one winning lipid at the end for that kind of application. So we do have quite a decent number of lipids in the library, along with some proprietary compositions. Now, at the end, I would like to thank the entire R&D members of Precision NanoSystems, both past and present, for all this work. Without their help, it would be definitely impossible to get all these data. And I would also like to thank for Professor Robin Shattuck, his group, and Anna Blackney. That was a fantastic collaboration with Imperial College London. So that was the fantastic collaboration on the vaccine applications. And I would like to thank you all for listening to me, and I would be happy to answer any questions that you have.